Every generation is to get a little bit further back from what was taken. And right now, of course, it's our language and culture and our history being taught.
Somewhere in a small village high in the Andes, a doctor draws a vial of blood from a patient and already knows, before the lab even runs the test, exactly what the result will be. Type O. Not because of luck, not because of a coincidence, but because in that community, it has been that way for thousands of years. Walk into a clinic in the Amazon, or a reservation in Montana, or a highland town in Peru, and the story repeats itself again and again. Entire populations spread across 2 continents and thousands of miles, sharing one blood type almost without exception. For more than a century, this pattern has sat at the center of one of biology's strangest and most stubborn puzzles. Why would one blood type, out of 4 possible options, take over so completely in some of the most isolated corners of the Americas? And why, after all this time, are researchers only now starting to piece together a real answer? This is the story of type O blood, the people who carry it, and the long scientific chase to understand where it came from. To understand why this mystery matters, it helps to understand what blood type actually is. Human red blood cells are coated in tiny molecular flags called antigens, and the ABO system sorts people into 4 groups based on which flags they carry. Type A carries A antigens. Type B carries B antigens. Type AB carries both. Type O carries neither. That absence is not a small detail. It is the entire reason type O blood is prized in hospitals worldwide as the universal donor, since a transfusion recipient's immune system has nothing on the incoming blood cells to react against. Type O is common everywhere on Earth, but in most populations, it shares the stage with A, B, and AB in a fairly balanced mix. What makes indigenous populations of the Americas so unusual is that, in many communities, that balance disappears entirely. Researchers studying indigenous groups from the Arctic down through Mesoamerica and into the deep south of the continent have repeatedly found frequencies of type O blood approaching, and sometimes reaching nearly total saturation. Numbers like these do not happen by chance in a normal, freely mixing population. Something specific, something historical, had to have happened. The leading explanation scientists have built over the last several decades starts with migration. Genetic and archaeological evidence points to a wave, or a series of waves, of early humans who moved out of northeastern Asia and crossed into the Americas during the last ice age, when lower sea levels exposed a stretch of land connecting Siberia to Alaska known as Beringia. This was not a quick crossing. Genetic studies suggest that ancestral populations spent a long stretch of time, possibly thousands of years, essentially paused in this region before pushing further south, a period researchers sometimes call the Beringian standstill. It is during this isolated pause that scientists believe a very specific genetic change took root. Deep within the ABO gene, researchers identified a particular mutation, a single change in the genetic code, that appears almost exclusively in indigenous American populations and is essentially absent from other groups descended from that same Siberian ancestry, including neighboring Arctic populations. In other words, whatever happened, it happened after the split in a small founding population that later gave rise to nearly everyone living in the Americas before European contact. If that founding group was small, and if it carried mostly this one genetic variant, the effect could spread through the entire population simply through the mathematics of genetic drift. A small starting gene pool, isolated for millennia with no new genetic material flowing in, does not need a dramatic explanation to end up dominated by one variant. It only needs enough time and enough isolation, and the Americas offered both in extraordinary amounts. For thousands of years after that initial migration, the peoples of the Americas developed largely apart from the rest of the world. There was no steady flow of new arrivals bringing different genetic material, no repeated waves of migration mixing fresh variation into the existing population, at least not on a scale that would meaningfully dilute what was already there. In population genetics, this kind of prolonged isolation acts almost like a seal on a container. Whatever genetic traits are inside stay inside, and whatever traits are outside stay outside. Over enough generations, a trait that started out simply common, rather than universal, can become the overwhelming default. especially in smaller, more isolated communities such as those in mountainous regions or dense rainforest, where population sizes were naturally limited and contact with outside groups was rare. This is part of why researchers see some of the highest recorded frequencies of type O blood in exactly these kinds of settings, from high-altitude Andean communities to groups deep in the Amazon basin, places where geography itself enforced a kind of genetic isolation long after the initial migration was complete. Isolation and chance go a long way toward explaining how a single genetic variant could dominate a population, but many researchers have asked whether there was more to the story. Could type O blood have carried some kind of functional advantage that helped early populations survive their environments? Several ideas have been explored. One line of research looks at infectious disease. Because type O red blood cells lack the A and B antigens, certain pathogens that rely on those antigens as attachment points may have a harder time causing severe illness in type O individuals. The best documented example of this comes from studies on severe malaria, where type O blood is associated with somewhat lower rates of the most dangerous complications. Malaria was not historically a major force in the pre-contact Americas the way it was in parts of Africa and Asia, so this particular advantage is unlikely to be the whole explanation, but it does illustrate a broader principle. The absence of A and B antigens is not a neutral trait. It changes how the immune system interacts with the outside world, sometimes in ways that matter enormously for survival. Other researchers have looked at high-altitude adaptation. Since so many communities with extremely high type O frequencies live in demanding environments like the Andes, where oxygen levels are lower and the body has to work harder to deliver it efficiently, whether type O blood itself confers any measurable advantage in these conditions is still debated, and much of the strongest evidence for high-altitude adaptation in these populations actually points to other genetic changes related to oxygen processing, rather than blood type directly. Still, the correlation is intriguing enough that scientists have not ruled out some interaction between blood type and the broader suite of adaptations these communities evolved. For most of the 20th century, researchers were working with fairly broad strokes: overall population frequencies, family studies, and comparisons between modern communities. What has shifted more recently is the resolution of the tools available. Advances in ancient DNA extraction now allow scientists to pull genetic material directly from centuries-old and even pre-Columbian remains, rather than relying only on blood samples from living descendants. This matters enormously because it lets researchers ask a much sharper question. Was type O blood already dominant in these populations before European contact, or did later historical events, including the demographic catastrophe caused by colonization and introduced disease, somehow shape or exaggerate the pattern we see today? By comparing ancient remains from North and South America against modern indigenous populations and against Siberian source populations, researchers have been able to trace the specific genetic variant behind the dominant American form of type O blood back through time with far greater precision than was possible even a decade ago. The picture that has emerged strongly supports the idea that this variant was already present and already common in the founding population that crossed into the Americas, rather than being a product of later events. That single point closes off decades of competing theories that leaned more heavily on post-contact population collapse as the primary explanation, and instead places the origin of this pattern squarely in the deep past, tied to the original migration and the long isolation that followed it. One of the more remarkable outcomes of this research is that the specific mutation behind the dominant American type O variant has become something scientists call an ancestry informative marker. Because this genetic signature is essentially unique to indigenous American populations and their direct descendants, its presence or absence in a DNA sample can help researchers, and sometimes individuals seeking to understand their own family history, trace connections back to that original founding population. For genealogists and population geneticists working together, this has turned a simple blood type into a kind of genetic compass, one small piece of evidence among many that can help reconstruct how ancient communities moved, settled, and remained connected across a continent for thousands of years. It is worth being careful here, though. A single genetic marker never tells the whole story of who someone is or where they belong. Ancestry connects to biology, but identity connects to community, language, culture, and lived history, none of which show up in a DNA test. Genetic research can support and, in some cases, confirm oral histories that indigenous communities have carried for generations, but it does not replace them. And the best research in this field has increasingly been done in partnership with the communities being studied, rather than simply on them. The story of type O blood does not exist in isolation from the larger, ongoing effort to understand how humans populated the Americas in the first place. For a long time, the dominant model was fairly simple: a single migration across Beringia, followed by a rapid spread south. Genetic evidence gathered in recent years, including the work on blood type, has pushed researchers toward a more complicated picture involving multiple migration pulses, a prolonged standstill period in Beringia, and possibly more than one route south, whether by an interior corridor or along the Pacific coastline. Findings from Siberian archaeological sites near ancient lakes and river systems have added more pieces to this picture. Since these regions appear to have served as long-term population centers and crossroads where different early human groups met, mixed, and eventually contributed to the ancestry of people who would later cross into the Americas. Separately, genetic studies have also confirmed unexpected contact events much later in history, including strong evidence that Polynesian voyagers and indigenous South Americans interacted directly, likely through Pacific voyaging, centuries before European ships ever reached the Americas. None of this rewrites the type O story on its own, but together these findings paint a picture of ancient human movement that was far more dynamic, resourceful, and interconnected than earlier, simpler models assumed. Understanding the genetic history behind type O blood is not just an academic exercise. It has real consequences for medicine today. Type O blood, particularly the O-negative subtype, remains the most urgently needed blood type in emergency medicine worldwide, because it can be transfused into patients regardless of their own blood type when there is no time to test. Communities with historically high rates of type O blood are, in a sense, sitting on a valuable medical resource. Yet many of these same communities, especially remote indigenous populations, continue to face real barriers to healthcare access, inconsistent blood donation infrastructure, and supply chains that do not always reach them when it matters most. Understanding the deep genetic roots of this pattern does not fix those structural problems, but it does add urgency to closing the gap between where this blood type is most concentrated and where it can actually reach the people who need it. There is also a growing body of research linking blood type more broadly to disease risk, including studies suggesting type O may be associated with somewhat different risks for certain cardiovascular conditions and clotting disorders compared to other blood types. As this research matures, it feeds into a larger shift toward more personalized approaches to medicine, where a patient's genetic background, not just their symptoms, informs how doctors assess risk and choose treatment. None of this means every question about type O blood in the Americas has been answered. Science rarely closes a book completely. It tends to replace one set of open questions with a more refined set of new ones. But compared to where researchers stood even 15 years ago, the picture today is dramatically sharper. The dominant explanation is no longer a vague gesture toward isolation and chance. It is a detailed evidence-backed account. A small founding population carrying a specific genetic variant crossed into the Americas during the last ice age, spent a long period effectively cut off from outside contact, and passed that variant down through countless generations with little to dilute it, aided by geography, isolation, and possibly a modest survival benefit against certain diseases. Ancient DNA has allowed scientists to test this story directly against real evidence from the deep past, rather than relying only on inference from modern populations, and so far, that evidence keeps pointing in the same direction. What started as a curiosity about blood typing charts has turned into one of the clearer windows we have into how humans first spread across, and then thrived within an entire hemisphere. The mystery of type O blood was never really about blood at all. It was about migration, isolation, survival, and the astonishing ways that history writes itself into the smallest details of our biology, details we are only now learning to read with any real clarity. Even with this progress, researchers are honest about the limits of what current evidence can prove. The precise weight given to disease resistance versus pure genetic drift is still debated, and different research teams continue to argue over how much credit each factor deserves. More ancient DNA samples from underrepresented regions of South America and Central America would help settle some of these disagreements, since much of the strongest ancient evidence so far comes from a limited number of well-preserved sites. There's also ongoing work exploring whether other genes linked to immune function and metabolism evolved alongside the ABO variant in ways that might have reinforced each other, rather than acting alone. What is clear is that this is not a closed case waiting to be filed away. It is an active, evolving area of research, one where each new set of ancient remains, each new sequencing technique, and each new partnership between scientists and indigenous communities has the potential to refine the story further. If there is one lesson in all of this, it may be that a question as simple sounding as why is this blood type so common here can, when followed far enough, lead all the way back to the Ice Age across an ancient land bridge and into the deep, shared history of an entire hemisphere of people. What do you think this story tells us about how much of our history is still hiding in our own biology? Let us know in the comments.